In this video, we're going to discuss the death of a red blood cell and how we end up recycling its parts. There's actually only 2 slides, other than these learning objective slides. There's only 2 slides, but it's full of information. So when the red blood cell dies, we want to know the fate of the heme group, which includes, of course, the porphyrin ring, as well as the iron, as well as the amino acids that are part of hemoglobin. And so really, when I say the fate of whatever, I really mean fate of hemoglobin. That's the main thing that we're looking at. We want to look at this thing called bilirubin. Where does it come from and what happens to it, as well as these terms ferritin and transferrin and what they mean. So remember that the red blood cell lives for about 120 days, and remember that it's got that flexible cytoskeleton, and which is really important because when you squeeze through very narrow capillaries, you need to have some flexibility so you don't pop and die. But what ends up happening over time is that as you become less and less flexible and we start squeezing through some of these places, it becomes increasingly difficult. So what we want to appreciate is that we've got this red blood cell, and the primary place that it's going to go to die is the spleen. Now, again, some liver and some red bone marrow contribute to that, but we're going to say spleen as the main thing. So what ends up happening is that in the spleen, there's actually a whole bunch of certain protein fibers called reticular fibers. It's in a reticular connective tissue loaded all up in the spleen. And these are really just tiny little, like almost like, if you remember what reticular fibers are, they're really very similar to collagen, but much smaller, narrower, but similar constitution. So the red blood cell is entering the spleen environment, and there's reticular fibers all over the place. So this is how the erythrocytes become trapped within the capillaries of the spleen. They become trapped because there's all sorts of reticular fibers all over the place, which isn't cleanly visualized here. So what happens from there is that, they get caught there, and things like macrophages, which are immune cells, will go ahead and start chewing up that red blood cell when they're stuck. Now, again, keep in mind that blood is always flowing through the spleen. It's just that when you're young and flexible, you can make your way through these kind of mesh networks between the spleen, and you enter in and you make your way out with a piece of cake. But when you're older and stiffer, you get stuck there. So in many cases, the red blood cells will pop and die. In other cases, they get stuck there, and they're digested then by these macrophages. So what we really care about then is that let's recycle what we need to recycle. So the question I have up at the top is, what do we want to save? Well, remember that red blood cells are really nothing but big old bags of hemoglobin. So the only thing we really have in red blood cells that's worth saving is the hemoglobin. So then the question is, okay, great, what do we do with the hemoglobin? So we see in this slide, hemoglobin is broken down into its parts. And what are its parts? It's broken down into the amino acids, because remember those polypeptide chains, and it's broken down into the heme group. Well, what is made up in the heme group? It's made up of the porphyrin ring and iron. Now, this is a good time to go back and review the makeup of a red blood cell. And remember that it's nothing but a big old bag of hemoglobin. So what is a hemoglobin? Or what is hemoglobin? It's a whole bunch of polypeptides, 4 polypeptides but containing also a heme group, which is the porphyrin ring and iron. So that's the makeup, right? So amino acids, porphyrin ring, and then iron. So this is what we're really interested in recycling. And that's what we're going to get into. So what do we do with the amino acids? Well, we take the amino acids and we break them down. We take the protein, break it down into amino acids, dump them back into blood, which eventually makes its way back into the bone marrow, and we will recycle them. Remember, these amino acids are just building blocks. So, amino acids can be used to make one type of protein, but when we chop it up, we can recycle those amino acids just to make another protein down the road. We don't need to dispose of them. We don't need to get rid of them. And so what is the fate of the amino acids? We recycle them. So there you are on the spleen. They dump back into the blood. They travel through the blood, go back to the bone marrow, and we use them to reconstitute hemoglobin when we're making new red blood cells. So yay, we've knocked that out. We know what happens to the amino acids. The answer is, describe the fate of hemoglobin's amino acids. We recycle them. Okay, there you go. Simple and easy. The next thing then is talking about this heme group, again, which keep in mind is made up of the porphyrin ring as well as the iron. One more time, just to visualize, the porphyrin ring is this stuff in yellow. Iron is right there in the middle. So I'll just kind of give you the punchline here. The porphyrin ring we're actually going to dispose of. We're going to get rid of, and we're going to actually end up peeing it out and pooping it out. The iron, though, is this precious metal that we're also going to recycle. So we need to recycle that, and when we recycle, guess where it needs to go? It needs to go back to the bone marrow, right? Because the bone marrow, when it makes new red blood cells, is going to need that iron. So that's what we're going to do with iron. But that porphyrin ring, we're going to dispose of. So let's see how that is done. Oops, wrong slide. Let's go here. when you look at that heme group, the porphyrin ring, and I've got one more slide to show you besides this, but the porphyrin ring is going to be metabolized into bilirubin, and then bilirubin then goes to the liver, and from the liver it will be excreted. It'll be pooped out in this case. And the iron, what do? We recycle the iron. The iron actually needs a certain boat to travel through, and that boat is transferrin. So transferrin is the name of the protein that transfers protein. So here, ferritin versus transferrin. Ferritin is actually the storage form of iron, and we store that in the liver. And then transferrin is the traveling version of iron. Okay, so ferritin is how iron is stored. Transferrin is how iron travels with the boat. And so that's why it says here, iron is hooked up with transferrin, and then it travels off to the bone. So let's take a little bit of a closer look, and this is the only other slide what we're going to care about. So again, here is your red blood cell. Red blood cell dies. It undergoes phagocytosis through those macrophages, and from there we're going to break it up into the amino acid component. Right, this was the polypeptides, like broken down into amino acids, which are then recycled, and so we followed that all the way through. And next we have to follow the heme group. So the heme group is broken down into the iron as well as the porphyrin ring, which gets converted into bilirubin. So again, the porphyrin ring gets converted into bilirubin. This biliverdin step, we're just going to skip it. It's just another intermediate step. So it's broken down into bilirubin. So let me follow through the iron first. So iron then is sent over to the liver through transferrin. Remember, the transferrin is the traveling version of iron. And then it's stored in the liver as ferritin until it's needed again. And when it's needed again, it takes the ride with transferrin. It travels its way over to the bone. And then from there, that iron with the recycled amino acids, with some other help, including EPO, is then going to make new red blood cells. And then the circle of life continues, circulating for, again, 120 days. So now here's where we're at. We've traveled. We've taken care of the how do we handle the proteins. We've taken care of how do we handle iron. The only thing that remains is how do we handle the porphyrin ring. So picking up there again, the porphyrin ring is converted into bilirubin. Bilirubin then travels to the liver for excretion. Now what ends up happening, we'll look at the liver in more detail in the GI system, but the liver will actually take that bilirubin and it will secrete it into the small intestine. Now, some of that bilirubin from the small intestine will travel through and eventually gets pooped out. You see here it says feces. And this conversion into urobilinogen and stercobilin, I don't need you to memorize that necessarily, but I do need you to see that some of that bilirubin ends up being excreted in the feces, in the poop. But interestingly, some of that bilirubin actually ends up getting reabsorbed back into the blood. Now, I know that sounds crazy because you might be thinking, whoa, you said it's waste. So I understand if the liver dumps it into the intestines and I poop it out, I get that. But then why am I going to bring some of it back into the blood? And the reality is there's no good reason for that. As we look at some of the things in the GI system, we might see that it's kind of almost an inadvertent reabsorption that occurs. But when that does end up getting reabsorbed, kind of inadvertently, we end up converting that into urobilin. And again, I don't need you to memorize the name urobilin. I need you to just know bilirubin. But that urobilin then is excreted in the urine. So again, I know it seems weird. You're about to go out this exit door, and a lot of it exits out the GI system, but some of it gets reabsorbed into the blood, which then gets peed out instead. And so at the end of the day, all these things are eliminated. So again, I know it doesn't make good conceptual sense for some of it to get reabsorbed, but just trust that it does eventually get excreted. And some of that is kind of an accidental reabsorption that occurs, and we've just found ways to manage that. And again, some of that will make sense when we talk about the gallbladder and liver and the GI system and how we end up recycling bile salts and stuff. In any case, that is the death of a red blood cell. And so one more time, just to rehash, when we look at a red blood cell, what is it that we need to handle? We need to handle the hemoglobin, because that's all that's in there really. We take that hemoglobin and we know that the polypeptides, the amino acids, are recycled. Fine, which then leaves us with the heme group. The porphyrin ring is converted into bilirubin, which is then excreted, some of which through poop and the other of which through pee. And the last is the iron. Iron transfers, riding through transferrin, into the liver to be stored until an appropriate time. It's stored as ferritin, and then from there, when the time is right, it hooks up with transferrin again to travel to the bone, which then remakes red blood cells under the influence of erythropoietin. So Have we tackled all of these? Describe the death of red blood cell. What is the fate of the heme group? Porphyrin ring, becomes bilirubin. Iron is recycled, and the words transferrin and ferritin are important there. Amino acids are also recycled. What is bilirubin? It's a waste product that comes from the porphyrin ring. Where does it come from? The porphyrin ring. And what happens to it? It gets peed out and pooped out. What is the difference between ferritin and transferrin? Ferritin is the storage form of iron, and transferrin is the traveling through the blood form of iron. And that'll wrap it up for this video.